Perfect. I think that we're um, ready to get going. It's a Thursday evening and week two of the course. By the end of this week, we've actually gotten to the through the kind of I guess, 1/4 of the semester. So it's all moving forward pretty fast. What we had lined up this week for you was really kind of more of a review type of a week. So this week we kind of revisited some of the really essential core themes that are going to be fundamental as we move forward in the A&P studies. I know that some of that was very, very familiar. Some of it might have been a little bit of an extension. So for that reason, we were actually able to cover, I believe, 3 different chapters. So I'm just quickly going to jump from this view to our Canvas course so we can see a nice view of what the week entailed. So just give me a second. I'll share another screen. And really here is a view of what I was hoping to cover this week, or what we've been working on this week. So we had a look of chapters 2, 3, and 4. And like I said earlier on, largely these would have been revision of the stuff that you've already covered in your prerequisite classes. So I provided kind of a nice little summary videos of each one. And you, of course, have your textbook and PowerPoints to help you. For this week, one of the biggest, or two assignments that I'm asking you to do are really to do the quiz for chapter 2 and quiz for chapter 3. There's no practice anatomy for either of those chapters, and I had no questions about the chapter 4. So I thought that that's a fair amount of work, and it's kind of a good way to make sure that we're all starting at the same place when it comes to the very basics that are going to be vital for your subsequent A&P studies. I also have some additional handouts and some additional material available for you. Earlier this week, I did already, actually yesterday afternoon, provide one of the review sessions during which I went through quite a bit of lecture materials. I didn't have anyone joining me in live setting for that, so that recording is there for anyone who's eager to watch it. But it really was kind of me just going through the highlights of the chapters 2, 3, and 4. And if I jump back to the PowerPoint that I prepared for tonight, you'll be able to see that I had quite a few slides for covering that material, and it was a little fast-paced, but I think that since it's a recording, it's a good resource for everyone to have available. One of the things that I've been working on during the past few semesters has been just checking in with the students, and there's never any pressure on students to share if they don't feel comfortable, but I always think it's an important thing that I'm very clear and I'm very transparent that these courses are not meant to be the same course from one semester to another. These courses are meant to kind of evolve and meet the needs of students who are taking them. So if there's specific things that we need to tweak, I'd rather tweak them during the semester so that we can make the changes and implement those changes so that it affects those students who have requested the changes. I think what we should do, we should just start with that quick check-in. I'm gonna jump to the actually second set of questions that I have, is that if you have anything in particular that you feel like you want to share about what's working well in this class, and also what could work differently, what could we do better, feel free to just unmute yourself and share that, or if you prefer to share it on a chat, or if you don't have any strong feelings either way, that's okay as well. I totally understand. It's probably been a busy week, so I can see if no one has any thoughts about that, that's totally fine as well. But I just want to make this space really quickly at the start to make sure that if you have something burning on your mind that you want to share, either about what we should keep doing or what we should change, feel free to do so. Obviously, even afterwards, I always welcome those comments. And I'm just going to give a few more moments. I know that we teachers have a tendency of kind of moving ahead really fast, not giving that space for quiet. Well, I'm not hearing anything burning in the mind of people, so I'm just gonna keep going through with what I had lined up. But at any stage, again, feel free to just jump in, and I'm also happy to pause the recording if something comes up later on. Really quickly about the weekly meeting times, I'm noticing that the Wednesday afternoon is a little quiet time. I'm happy to still offer sessions there, but if it looks like that's not very popular, we might have a look if we should schedule that meeting time for another time. So I'm putting it out there that if a time doesn't work, I'm happy to change them around as well. But I don't have a problem also making just a recording if it helps anyone. Like I said, this week I'm asking you to complete those two quizzes, quiz for chapter 2 and quiz for chapter 3. And as with the last week, both of those are completed at the Connect, and then those grades from those assignments will automatically transfer to the Canvas course. Both of these assignments are due at the end of the week, so Sunday night, I believe it's 1 minute before midnight, and both assignments are worth 5 points. I wanted to kind of, even though I know that we're still working on this week's material, I wanted to kind of draw our attention for what's happening next week so that everyone can kind of plan accordingly and have an idea of what's to come. Next week, we're actually moving to our first system that we're having a look of. So we'll have a look of chapters 6 and 7. And I do have a quiz for both of those chapters, and I do have a practice anatomy assignment for both of those chapters. In addition to that, I also have this food for thought assignment, which is really just a really brief, short answer, maybe 3-4 sentences long. I'm very flexible how students want to take part on that, but kind of a chance to apply the content that we're covering next week. So next week, we'll be looking largely at the integumentary system, which really covers the skin, hair, nails, glands, those associated structures with the skin. So the food for thought is all about goosebumps. You'll get to learn a little bit about what causes goosebumps and what they are and what their purpose is and so on, and then just kind of summarize your thoughts about those. Another thing that's happening next week is that we'll be taking our very first exam and practical. And these exams and practicals are actually taken fully on Canvas, and the first exam will cover chapters 1, 6, and 7. So some students sometimes find it easier that they work through the weekly coursework and then tackle the exam. Practical covers the same chapters. In terms of the exam is going to contain questions that you have already faced and met as part of the quizzes for those chapters. So it's not always word for word same questions, but largely it is, and largely it's very, very familiar to that. Practical also covers the pictures that you would have seen on the practice anatomy. Obviously, there's not going to be every single question that you've done for your practice anatomy, but there's going to be some of those questions. So just wanted to put that on the horizon for the next week so you know what to expect. So I kind of assume that next week might be a little bit busier week ahead. So having said all that, I think that we should focus on this week's material. And this is the time where I'm really open for this session to kind of take any shape or form that you wish. This is to serve those people, especially who are able to attend, so that you can express what would be most helpful for you. During the Wednesday's meeting, when I was kind of here by myself, I did actually record a review of all those 3 chapters that we had for this week. So chapters 2, 3, and 4. I did it in a little bit longer than an hour, but I covered pretty much all the key points of each of those chapters. I'm happy to do it again. But also, if you kind of feel like you have a good handle on those, I'm also happy to skip that because that is available as a recording for anyone who wants to watch those. Sometimes some students also prefer that we go through the homework, so it kind of gives them confidence of that. So I'm very open for whatever you guys feel that would be most beneficial for you. So if you have some preferences or hopes or requests, feel free to just unmute yourself or drop them to the chat. If nothing comes up, I have a couple of ideas, but I really want this to be serving whatever is going to be most useful for you. Again, I'm just giving a few more moments if anyone has any particular views about what would be useful for you in terms of what we want to cover.
I'm okay with trying something different since you recorded already.
Sounds good. It gets a little repetitive if I do a recording of all of the chapter materials time and time again. If you're okay with that,
Yeah,
that would be happy.
I'm good with it. I don't know about anyone else, but I'd be okay with it.
Perfect.
Yep.
Let's.
Oh, that would be really good.
Okay, perfect.
Thank you.
Let's make you watch the same stuff all over again. So I want to show you really quickly, if that's okay, one question that kind of keeps coming up sometimes in the quizzes. And this is by no means saying that it wouldn't be a good question. I actually agree with the question because, like I said, Connect has been designed for particular type of learning, but I don't think that it's perfect at all settings for everything. So let me share my screen of a couple of questions that keep coming up. So sometimes on these quizzes, I have questions where there are multiple correct answers, such as I think that this was a question. Let me just make sure that I'm not saying a wrong question. Question 16 on your this week's chapter 2 quiz. So when there's multiple correct answers to the quiz question, it's easy to get kind of confused about what the feedback actually tells you. And you know that I'm all about look through the feedback that you get on the quiz if you want to retake the quiz. So when you have this kind of a feedback where you have tick marks on green showing that something went correctly, and then these X's on red, that tells that it didn't go correctly. So in this particular question, I believe that the correct answers would have been chemical energy and potential energy. So you'll notice that the student clicked chemical energy. So on the feedback, it shows it as a correct answer. Potential energy was the second correct answer. And because the student didn't click it, it's showing up on the feedback as a kind of a wrong selection. So with that red cross. You'll also see on the feedback that the not choosing the kinetic energy shows up as a correct answer. So that's why we have that green tick mark on that. And for the thermal energy, since that was not something that we were wanting you to click on, it's showing up as a wrong answer since it was chosen. But I realized here's a second view of the same question again. And I realized that it can be sometimes kind of almost like decoding what is the software trying to tell you. And that's especially when there's multiple correct answers to the question. So we see here the chemical energy would have been correct, but potential energy was what we were looking for to be chosen. Kinetic energy was chosen, but shouldn't have been chosen, and it was a correct choice not to choose the thermal energy. So I just wanted to put it out there, just kind of as a quick disclaimer, because a lot of students are kind of finding that tricky, and I completely understand why. So you know how to read that feedback that you get back. Since you are here, I want this to be super worthwhile for you. So I'm going to be completely honest and blunt about it and tell you that I really, if I was a student and I would be trying to figure out what's the best way to take this course, I would work through all of those quizzes and I would take notes wherever I get something wrong, and then I would retake until I get good amount of points, because those points are really what adds up super quickly to your final grade. I also would be very wary about or aware of the fact that your exam contains exactly similar questions that you've already seen on the quizzes. So if you wish to somehow make notes of those quizzes, whether it's you screenshotting things or printing things or just doing your own things, you do what works for you. But remember that all of our exams are open notes. So really doing the homework is you preparing to do really well on the exam, and same goes for the practical as well. So that was one thing that I wanted to put out there to the universe. What I would propose that we will do, if that's okay, and again, feel free to just shout out if you have different thoughts, is that let's go through the homework. That's really probably the most useful way. We kind of get to review some of the material, but you also are having hopefully a little bit of a help towards getting those points collected. So I was going to start with the chapter 2 quiz. You'll see that it's 16 questions, all adding up to 5 points. And the first question that we're seeing here is a question where you'll be dragging these boxes to show different kinds of bonds that we have going on. And what we're seeing here, we're seeing water molecules made of 2 hydrogen, 1 oxygen. So that'd be the first thing that you would label that, hey, we're seeing a water molecule here. We also learned during this week, and I think that you've learned in your previous prerequisite courses, that there's different kinds of bonds. So bonds are a way how atoms are formed by different, or molecules are formed by different atoms joining together in various different ways. And here we see 2 different kinds of bonds. The first bond that we see is a covalent bond. Covalent bond is formed when 2 atoms share electrons. It could be more than 2, but in this case, it's actually 3 different atoms, but it's the hydrogen and oxygen that are sharing electrons. So electron of the hydrogen and oxygen are shared so that both of them reach the state of having a full valence shell. The electrons don't fully move from one atom to another, they're just shared. And there's, whenever we share electrons, a pair of electrons, we show it on these diagrams as this line between these atoms. So there's 2 shared electrons that we're seeing here between each hydrogen and each oxygen. In addition, we ended up seeing that water was so special for life that actually the bonds that water molecules form with other water molecules had a special type of bond that we talked about. And to understand this hydrogen bond that we see between water molecules, we have to recognize that actually these electrons that we're seeing shared between hydrogen and oxygen are not shared evenly. A lot of the times those electrons would be shared evenly, but in this case they are not. In this case, these electrons that are shared between hydrogen and oxygen, they spend more time at that oxygen end of this molecule, so there's a slightly greater negative charge. Remember, electrons were negatively charged, so there's more negative charges at this oxygen end, and while they were spending a little less time at the hydrogen end, there's slightly lesser negative charge, so there's slightly positive charge at this hydrogen end. And we remember from the chapter material that opposites always attract each other. So each water molecule is slightly negatively charged at the oxygen end and slightly positively charged at the hydrogen ends. And that means that there's these weak hydrogen bonds between oxygens and hydrogens. And this is really the explanation why water kind of tends to stick together when we look at individual water molecules. They form this cohesion between different water molecules. So that was kind of an in-depth explanation of that, but I think it was a word of seeing. At any stage, if you have questions, just feel free to jump in and shout out. Here we're seeing functional groups. And when we talk about functional groups, these are an important part of studying the chemistry of materials, because these functional groups always behave in a similar way, no matter in what atom we end up seeing those. So here we're asking you to classify the functional groups based on the type of molecules where we typically would find those. So first of all, we're seeing hydroxyl group, which are typically found in sugars and alcohols. We're seeing an amino group, which we typically find in proteins. So remember, proteins were amino acid chains. And then we have methyl group. Methyl groups are often found in fats, oils, steroids, and then we see phosphate group. Phosphate groups are typically found in nucleic acids. So remember that, for example, DNA and RNA, the basic molecular structure contained one sugar, one phosphate group, and then the variable base group. So the phosphate and sugar are always found on nucleic acids such as DNA and RNA. And then carboxyl groups are typically found in various different kinds of sugars and proteins. So that's something that's characteristic for those. So I hope that that kind of helps. Really, I provided a really good handout of these different kinds of functional groups on your additional material on the learning module, if that's something that you find that helps you to learn from that. Let's have a look of the next question, which asks you again to move these boxes to appropriate under appropriate headings. So we're having a look of carbohydrates, proteins, and lipids. So examples of carbs would include glucose, sucrose, monosaccharides, and cellulose. So these are all really the most accessible form of energy that the body uses. Typically, we always gravitate towards carbs as a source of energy in the body. We'll end up finding that the simple carbs such as sucrose, are used first, and then the more complex carbs, it takes multiple steps to break those down. So that's why with sugar, you really get that sugar rush. You get quickly released energy, but it also gets burnt off quickly, and then that energy is gone. Whereas with complex carbs, they stay around for longer for when you might need that energy. Let's have a look of next, let's go to lipids. So lipids are the second most easily used form of energy in the body. So if there's no carbs available, your body gravitates towards using lipids as a source of energy. So this is where those low carb diets are based on, that let's burn off fat cells by not giving body any carbs, so that you would go towards that. There are some dangers with that approach, but in a controlled setting, that might be a reasonable diet choice. So lipids contain fats, and you remember that triglycerides and fatty acid chains were kind of the backbone of when we're starting to look at different kinds of lipids. There is a third group, eicosanoids, which is really a group that we don't fully have a grasp on yet. So I anticipate that this space will change in the textbooks in the years to come. But at this point, if you're able to classify it as under lipids, that's a good place to be at. And finally, if we look at proteins, proteins are the kind of the third and the least preferable source of energy in the body in terms of the accessibility of the energy. So proteins were chains of amino acids, and we end up seeing that proteins could be studied in many different levels, and the quaternary structure would be one of those levels. So we see levels from 1, 2, 3, and 4 that we can look at, and the complexity of the protein in terms of its visualization gets more and more complex as we move on to the higher level structures. So that was what I had on this question. I'm just gonna keep going, but at any stage, if any questions, any comments, feel free to jump in. Here we have 3 different statements that we're asking you to complete by dragging the correct word there. The first statement tells us that a large 20 metric ton rock sitting on top of a hillside would be an example of potential energy. One way to think of a potential energy is that we're really looking at a force, energy that's stored in an object thanks to us moving against the gravity. So me lifting this pen up stores more and more potential energy on this pen, and when I let go, that potential energy is then released and turns into kinetic energy. So anyone who has ever helped their friend to move on to a house that has more than one floor or more than the ground floor, and for example carried washing machines or fridges or something like that, knows that potential energy that gets stored into that object as you're moving away from or moving against the gravity, it does take effort to move things. So that rock holds energy. That energy is not really released, but it's stored there. And if we're letting that object then move towards the gravity, that energy is released. The next one that we're seeing here, to free the rock from the top of a perch, you need a catalyst, which could take the form of a person or a leveler to free the rock. In this case, really, the catalyst is the term that we're wanting to show you. That's something that kind of triggers that change. Just simply a book on a bookshelf that has potential energy, since it's stored high up there, it's just not going to randomly fall. There needs to be some sort of a change that triggers that fall to happen. So that change would be the catalyst. Once freed from atop of the perch, the rock demonstrates kinetic energy as it plummets to the valley floor. So like we saw with this pen, it has potential energy while it's resting against the gravity. But when we're seeing kinetic energy, we're talking of energy in motion. So whether I just let go of the pen or whether I throw it across the classroom, it flying, moving through the air would be an example of kinetic energy. So a good example of that is, for example, an engine of a car that converts chemical energy stored in a gas to kinetic energy, which is actually then we're moving that object. And final example that we're seeing here, or statement that we're seeing here, is that in the process of rolling down the hill, work was done to push aside trees and other stones that stood on the way of the rock. So really work is a term that we're looking at here. We're doing some kind of a change, we're moving, we're causing a change in terms of that. So those were the words that were kind of the only ones that fitted into these questions. So I hope that that kind of makes sense. Let's have a look at the next one. On the next one, there was a little bit of an animation to look at first, and then we have a couple of questions about enzymes. So when we talk about enzymes, I'm gonna show you an illustration from my slide deck. Actually, I think that that's gonna be easier. So bear with me as I pull up the slide deck. And this was in that presentation that I recorded earlier on. So I'm just gonna show you the highlights. So now we're talking about chemical reactions that would happen. And these chemical reactions would happen as it is normally. So in this example of a chemical reaction that you'll see on the screen, I have this green block. And all that's going to happen in that chemical reaction is that one large green block, let's call it a molecule, gets broken into two smaller molecules. That's the reaction. And this reaction would happen even normally. So there's really nothing that wouldn't happen in terms of that without the enzyme. This reaction would happen normally. The larger molecule would be broken into smaller molecules. But what enzymes do, they help in reactions. And how they help, there's many different ways what that help can, what form of a shape that can take. It might be speeding up the chemical reaction. It might be making a lower activation threshold or less energy being needed, or making it more complete reaction, or whatever it is. So in this illustration, the enzyme is this yellow particle. And what we call this green block is the substrate, and the substrate binds to the active site of the enzyme, as you see on that middle picture. So it binds to this active site of enzyme, and that can cause sometimes the enzyme to actually change in shape. And the enzyme, it helps this reaction to happen. This reaction would happen anyways, but enzyme just helps it to happen. And I'm going to use in quotation marks better, whether it's faster, easier, more efficiently, with less energy, more completedness. And once it has helped this reaction to happen, the enzyme returns back to its normal shape as the substrate leaves the active site of the enzyme, and enzyme can be reused time and time again. So that's what the question that we were seeing. And let me jump onto that screen. Now we should be seeing the screen. So when we were talking about that green block, that would be the substrate that bound to the active site of the enzyme. I think that the most common enzymes that we see are catalysts. Catalysts are a special type of enzymes. They speed up chemical reactions, but there's many other kinds of enzymes as well. So that's really what I was wanting to ask on that question. Let's have a look at the next question. Here we're matching chemical elements with their chemical symbols. So I provided you a table of that shows all of the periodic table of elements. So that's a good reference, but it's going to be important for us to know what these letter abbreviations mean. So we're seeing that oxygen is coded in chemical shorthand with an O. Carbon is coded in chemical shorthand with C. And hydrogen is coded in chemical shorthand with H. So you often see C and H being used in organic molecules because really it is the hydrocarbon backbone that all organic molecules are made of. We also see Cu for copper, and we see Co for cobalt. These are really things that more you use different kinds of elements in chemistry, you just become familiar. You can always look them up. Typically, most of these abbreviations either come from English or Latin names for the materials. So that's really the explanation why it sometimes doesn't make sense. For example, I'm trying to look here an example. Let's do the copper. Cu doesn't really sound like a copper. It comes from the Latin word for that. So that's the answer to that question. Let's have a look at the next question. Here we're being asked to identify statements regarding DNA. So DNA is really what stores our genetic material in each cell. At the nucleus, there's the DNA. So DNA contains coding for everything in your body. Even though that cell wouldn't be actually showcasing or using that gene. So gene was a segment of the DNA that coded for something useful or trait or a protein. Those all are included in the genes. So I often talk about, when I talk about DNA, I use a comparison that your DNA is like a cookery book. When you cook dishes, you rarely would take one cookery book and cook every single recipe that's there at the same time. You would choose different recipes at the different times. So you can consider that recipe as a gene for something. So in this case, the recipe codes, it gives us instruction how to make a particular dish. So similarly, our genes give us instructions how to make particular proteins or particular traits, but you never actually do all of them at the same time. You choose what's needed. So DNA gives instructions for synthesizing body's proteins. So proteins are really the final product that comes from the RNA, but DNA is where it's coded, and RNA is a copy of a particular segment of a DNA that codes for a specific gene so that it can be then translated into a protein. So I'm looking for answers that DNA codes for or constitutes of genes, and it gives instructions for synthesizing proteins in your body. Again, just to highlight this concept, I'm gonna jump to this slide deck to show you in case I kind of sometimes think that it's easier to illustrate, at least my mind works that way. So let's pull up an illustration. So here we're seeing behind this curved line would be our nucleus of the cell. DNA never leaves the nucleus. DNA always stays in the nucleus of the cell. So another analog that I sometimes make for my students is that I say that think of a book in a library. And this is a special collections book. So it's really, really precious. So they don't really let you take that book out of the library, but you can go to the library and read that book. You can have a look of that, or you could consider it kind of like a short-term loan or within only library loan book. So you're not allowed to take DNA out from the nucleus, but you can go and make a copy of the DNA. Similarly, in a library, you can go and make a copy. You can take your own notes. You can transcribe what's written in the book, or you can take photocopies of the book that you cannot take out of the library, and those photocopies you can take out. So RNA is almost like a photocopy, or almost like a copy of the section of the DNA that you were curious about. So this process of making a copy of a DNA or a section of a DNA is called transcription. Then the RNA can leave the nucleus, and once it gets to the cytoplasm and comes in contact with ribosome, here on this illustration ribosome is shown as this little blob. Ribosome helps to read the instructions of the RNA, which are essentially a copy of a section of a DNA, and combine different kinds of amino acids into a chain. And once you have a chain of amino acids, it becomes a protein. It folds into a protein. So we're really translating those instructions from the RNA to something that's useful and kind of gives us an actual concrete output of the coding or of the instructions. So I hope that that helped a little bit. So I'm going to again jump back to our quiz that we were seeing. So like I said, DNA constitutes of the genes and gives instructions for synthesizing proteins. Let's have a new look of the next one. RNA plays role in which of the following? RNA plays role in synthesizing proteins and copying instructions from DNA. So like we just saw, RNA is what could leave the nucleus to the cytoplasm. And we need to get to the cytoplasm because it is within the cytoplasm that ribosomes then give a meaning to that coding. So they're really copying. RNA is a copy of a section of a DNA that can be then used to synthesize proteins. So that's the only option of those that kind of make sense. The next one asks us which of the following statements is correct regarding biological properties of water. So we saw that water is really special for life. We really have an anticipation that most living forms evolved in water. Most living forms are largely made of water and require water to survive. There's few exceptions where certain things can survive periods of time without water, but that's why we're really looking for signs of water in foreign planets when we go there, because we don't think that life can exist without water. So let's have a look at this statement. This statement says that not only does water ionize many other chemicals, such as acids and salts, but water itself ionizes into H and OH. So what we're seeing here is that one of the unique characteristics of the water was that water is really good at breaking down, dissolving many things. And as it dissolves things, it can kind of impact the charges that are then resulted as a result of that process. And that all comes back to that chemical formula that we just saw for the water molecule. In the interest of time, I'm just going to keep going, but if you have any questions, feel free to just shout out or drop it to the chat. Let's have a look at the next question that we have here. So here we have a question which is actually a true statement, that catabolism consists of energy releasing decomposition reactions. So I'm going to try to use here a little bit of an illustration to show, and I'm actually going to do it on the PowerPoint because it might be easier. So let me jump there and make a slide for us there. And I'm going to share that screen with you. So you should be seeing a blank page on the PowerPoint. And there's two things that can happen. So let's start with that catabolism. And I'm sorry, I don't see the last of that word, but the first one that we saw was catabolism. What happens in catabolism is that we have this large molecule, and this large molecule contains many different kinds of bonds that hold smaller particles together. I think I can actually illustrate it even better. So let me just draw it differently. So here we have different kinds of atoms being held together, forming one large molecule. And you remember that whenever we had these bonds were a way of storing energy. So energy gets stored into these bonds that form large molecules. So now if we take this large molecule and break it apart into its components, in that process, we're breaking apart those bonds and we're releasing energy. So what catabolism does is that it releases energy as it breaks larger objects into smaller objects. So I often think of this in a way that in nature, what's natural to happen most of the time is that pretty much everything tends to decay and break apart. Let's take an old historical building. Over time, that old historical building will fall apart into separate bricks and mortar and so on and so on. And as it breaks apart, energy is being released. So in nature, we're really constantly trying to release energy. This is actually exactly what we see happening in your digestive system. As you're eating food, that food gets broken into smaller and smaller molecules and then to the very basic substances that it's made of. And as we're doing that, we're releasing energy for your body to drive processes. I know the question doesn't ask that, but I'm just going to highlight it to make it clear. Anabolism is an opposite reaction to that. In anabolism, we have individual atoms or small molecules, and by adding energy to this equation, we are actually combining these individual components to make something larger out of them. So this is what your body is doing when your body is building tissues or body is building different parts. So it all consumes energy to take these small things and put them together. It uses resources to do that. That's why we need to get energy to drive body processes such as building new tissues. And you don't build new tissues only when you're physically growing, you're constantly replacing tissues and so on and so on. So that's really what that question was after and what we're meaning with. Let me just jump back to the quiz. I want to make sure that I don't get too sidetracked, but I also do want to explain the questions that do come up. The next question that we're seeing here, there's again a little animation to watch, and then we're asking about the different parts of an atom. Atom has different parts, and this one asks, what is the positively charged part of an atom? And again, you won't be shocked that I have a great illustration of that on the PowerPoint slide deck. And let me just jump there. Here we're seeing an atom. This is a very schematic representation of an atom. So atom is like the smallest components that the matter is made of. And we see that atom has a nucleus that's made of protons and neutrons. These protons have a positive charge, whereas neutrons, as the name proposes, are neutral. They don't carry any charge. But around this nucleus, you can think of it almost like little planets circling the nucleus. We have electrons circling the nucleus, and these electrons actually carry negative charge. So this was what I was talking about, why water molecule, when it was sharing the electrons between hydrogen and oxygen unevenly, the one end became slightly negative because electrons moved there. So you'll notice that in an electrically neutral atom, there's the same amount of pluses as there are minuses, but sometimes we can end up with a situation where we have more pluses or more minuses. So we're changing the overall charge. So in that question that we just saw, we just wanted to make sure that you knew that protons were those positively charged particles. That's what we're really after with that question. So let's have a look of the next question here. Then, oh, I'm just gonna check, did I have more questions there? I also had a second question there. The second question tells you that number of electrons in the valence shell determines what type of bonds an atom can form. So again, kind of just really quickly jumping to that PowerPoint that I had prepared. So there are different number of electrons around different atoms, and really what we're trying to do, or what atoms are trying to do, is to have a full outermost shell at as much as they can. So we're only focusing on the outermost circle. But these circles can only fit in so many electrons, so the innermost, the first shell, contains 2, second 8, third typically 8. So atom always strives to fill their outermost shell. Sometimes it might be easier for an atom to get rid of one electron and release the next shell as its outermost one, and that way get that full valence shell. So really, by looking at the number of electrons, we're able to predict how reactive different atoms, different elements are, and a periodic table of elements shows the number of electrons in each of the substances, and especially from that we can figure out how many electrons are on the outermost valence shell, so we can predict that these reactive elements, reactive materials, are very likely to do chemical reactions, whereas these elements that have full valence shell, they're very stable and they're very unlikely to do chemical reactions. And that was a little bit of me jumping to explain in more detail. So we're seeing that it's true that the number of electrons on the valence shell, so valence shell meaning the outermost shell of an atom, tells us that what kinds of bonds is it likely to form. Let's have a look at the next question. Next question, again, is another type of way for us to see the chemical symbols of different elements. C for carbon, Mg for magnesium, Cl for chlorine, Na for sodium, Fe for iron, and K for potassium. And again, these came from mostly Latin and English names. So you'll see these being used and they will become more familiar. But feel free to use your textbook. Like I said, these quizzes are not something that you should just know. You will use your textbook to guide you. So on this question, next question, we're seeing a reaction. And in a reaction, we always have stuff going in to the chemical reaction. That's typically on the left-hand side of the chemical reaction. And then we have stuff coming out of the chemical reaction. And we call this material that comes out of the chemical reaction as a product of that chemical reaction. And again, I will showcase that concept on the PowerPoint. And like I said, I did a full kind of a recording explaining that. So the stuff going into the chemical reaction is known as reactants. And the material that comes out of a chemical reaction is the products. Typically, many chemical reactions are just moved to one direction, so they are irreversible chemical reactions. They just go to one direction, but we have sometimes chemical reactions where it can move back and forth, so they are reversible chemical reactions. So that was all I wanted to show, the products and reactants as it relates to that question of the quiz. Continuing onwards, we have statements and we're matching terms with that. These are all different kinds of things that are really related to water. So we see that molecules must be dissolved in water to move freely and react. That would be an example of solvency. So water dissolves many different things really, really well. That's why water is so fundamental for life. That's why we can use water to do many chemical reactions. Cohesion referred to the fact that we saw earlier on that these water molecules, individual water molecules, were attracted to other water molecules. molecules. So they formed these weak bonds, but these bonds help to keep the water together. If you've ever taken a glass of water and filled it, kind of overfilled it so that it's doming, it has this kind of a little blob on top of it, water just doesn't start flowing down. That's an example of cohesion, how these bonds that keep water molecules together help that to happen. When we're talking about that water lubricates organ surfaces and their surrounding area, we're talking about adhesion. So cohesion and adhesion. Adhesion is an example then how water molecules are attracted to other surfaces, especially polar surfaces. So when you go out after a night and you see humidity still stuck on spider web surfaces of the plant, leaves, and so on, that's an example of adhesion. Water molecules are attracted to other surfaces. When we talk about chemical reactivity, we're talking about that concept that water ionizes and can be incorporated into other molecules. We're talking about the fact that water, again, is able to be behind many different chemical reactions, breaking apart molecules, but also putting molecules together. And finally, the high heat capacity helping to maintain internal body temperature is a good example of thermal stability that's achieved with the water. So if you think about it, where people typically live in the world, people typically gravitate towards living on coastal areas, because wherever we have large bodies of water, water during the day binds the excess heat so it doesn't get too hot in that area, and then during the night it releases that heat so the temperatures don't drop cold drastically. So often when you live by a big body of water, the temperatures remain more constant. Here in Arizona, we see that we have huge night and daytime temperature fluctuations because we have so little humidity. So similarly, in the body, we can use water, and most of our blood is made of water, to transport heat from one body part to another. So for example, at gym, when you exercise, your muscles are doing work, converting chemical energy into a movement. And as part of that conversion, you remember whenever we converted energy from one form to another, we produce heat. Now, to prevent you from overheating as you're working out at the gym, we can transport that heat from those areas that are heating up with the blood to our body surfaces, such as skin, where we can get rid of that heat, for example, through the sweating and evaporation and conduction and other ways. Similarly, in a cold weather, your nose tip, your ears won't freeze because we're constantly, even if they're not covered, because we're constantly transporting heat to those with your blood that circulates to those parts. So we're kind of keeping them warm. And the last question in the chapter 2 quiz, we're seeing that question that we visited earlier on: energy that is stored in the bonds of a glucose molecule exemplifies which of the following? So glucose molecules are large molecules that store chemical energy that has also potential energy. So chemical energy, because we can break these chemicals, glucose molecules, into smaller and smaller, releasing energy from the sugar for your body to do its job, for example, as we see in the mitochondria that produce ATP, but it's also potential energy, because this potential energy, this energy that's stored in the glucose, can become something, but it's not becoming yet. It needs to be, something needs to happen for that. So it has this potential to become something, but it's not quite that yet. So that completed our quiz 2. I hope that that helped a little bit. I do know that we're at the full time, so I want to be really respectful of your time, and I don't want to keep you over time. For anyone who is interested, I am still happy to go through the quiz for chapter 3, and I'll keep up a little faster pace, but if you have to drop out, I completely understand it. I'll try to do a better job of managing the time, but I think that it is a worthwhile exercise for us to have a look of that quiz 3. So really quickly on here, we're starting with a question where you're completing sentences by dragging the correct terms. So cells are small typically, and there's a reason why cells are small. So cells are typically measured in scale of micrometers, which is 1/1,000,000th of a meter. So this goes back to when you learned about the metric system. So cells are typically size of millimeters or micrometers. We do have few odd cells that are longer. For example, in your spinal cord, you have long neurons that run up almost a meter in length, but they are exceptions. So cells are in the scale of micrometers. Cells can only grow to a certain size, in part due to the relationship between its volume and surface area. So there is an optimal size for the cell. And again, I'm going to illustrate that from my slide deck of the slides that I prepared ahead of the time before our class. So here we are seeing a cell. Let me find it. Provided that I find it. Really, I don't want to get sidetracked. So we were seeing the scale of the cells, and here we're seeing that cells have to remain small, because if the cell grows really large, then the ratio of the surface area of the cell, the amount of surface area that it has, becomes lesser than the volume that the cell has. So there's an optimal size where these two are in a perfect balance. One way how I explain it to my students is to think of, if a cell is really large, it's going to take a long time for materials to travel from here, from the edge of the cell to the center of the cell, if we need to transport materials that way. And because transporting materials consumes also energy, we might actually end up, if the cell was huge, consuming all that energy before it even gets to where it needs to get. So I compare it to a gas tanker that you have, or those gas transportation that you see on the road. It burns gas to move these gas tankers from different cities to another. We don't want to have such a long distance to go that there's no gas left in that tanker because we've had to travel such a long distance. So that's really the explanation for the ideal size of a cell. Let's continue with those questions. We're seeing an egg cell is one of the few human cells that can be seen with the naked eye. So typically with a naked eye, you can roughly, if you have really, really large cells, see the cell. Typically with a microscope, you can see the nucleus of the cell within the cell, but that's about it. And then you need an electron microscope to see the smaller stuff. But egg cells are so large that you can actually tell it with a naked eye without magnification tools. Since the volume of the cell increases faster than the surface area, if a cell grows too large, then it runs the risk of rupturing. So this relates to that discussion that we had before. If a cell would be really large, the volume inside would be putting too much pressure on the edges of the walls of the cell, and there's a risk that it ruptures. So another reason why cells typically are small. Kind of continuing through these membrane-bound organelles, that's the site of protein and lipid synthesis, endoplasmic reticulum. So again, I'm going to show it on the slide deck, what I'm talking about. Endoplasmic reticulum is really kind of an extension, if you wish, from the membrane that surrounds the nucleus. And endoplasmic reticulum is simply a network of these tubes and membranes within the cell. And there's two types of endoplasmic reticulum. We call smooth endoplasmic reticulum, that the surface is just smooth, there's nothing much there. And then we have rough endoplasmic reticulum, where we have little ribosomes attached. And you remember ribosomes were those structures that converted RNA into proteins. So literally rough ER looks like dotted on a microscopic scale. So that's what was meant with that question. So endoplasmic reticulum is the membrane-bound organelle. That's the site of the protein and lipid synthesis, because that's where we're making those proteins with the help of the ribosomes. Here you're labeling different parts, different organelles of the cell. So nucleus is typically located at the center of the cell, contains the DNA. So we saw DNA doesn't leave the nucleus. We talked about this endoplasmic reticulum. Smooth endoplasmic reticulum has many different functions. Rough endoplasmic reticulum contained those ribosomes. So this was the site where we made the proteins based on the instructions from the RNA. Golgi apparatus, Golgi complex is often something that I describe to my students as an analogous of a distribution center, of a trucking center. Trucking company. So Golgi apparatus is made of membranes that kind of wrap around each other. And what Golgi apparatus does, it receives different kinds of materials in it, it sorts them, repackages them, and sends them out to wherever they're needed. So you can kind of think of the Golgi apparatus as a distribution center of a cell. Lysosomes are an example of little sacs that have membrane around them that are inside the cell and help to really carry materials around the cell. So lysosomes in particular contain enzymes and typically digestive enzymes. So they help to move them. Those enzymes don't just float around free, they're enclosed into these sacs. Mitochondria instead is often described as a cellular powerhouse. It's where the ATP, which is the chemical form of energy that drives cellular processes, is produced. We now know that that statement is not 100% correct. ATP is produced in other parts of the cell, but still most of the ATP is produced at the mitochondria. And centrioles instead are these structures that play an important role when cell divides into two during the process of mitosis. So all of these structures, again, you can see on that presentation that I have recorded on Wednesday afternoon. I won't spend time on that now, but we're seeing here the Golgi apparatus. We saw the vesicles, we just talked about those. We talked about centrioles. And I'm sure I have somewhere there also about mitochondria, a little bit of information. Jumping back to the quiz, because like I said, I realize I'm a little bit over the time, so I want to keep going through it. Next question that we're seeing the endoplasmic reticulum, and you notice that we have smooth endoplasmic reticulum, which played many different chemical purposes. In this case, the one that has been highlighted is that this organelle synthesizes phospholipids. And then we had the rough endoplasmic reticulum, which was really the site where proteins were made, and proteins were made by ribosomes, these little dots on the surface of this rough ER. When we talk about cisterns, these are really just folded or flattened sacs of this network of membranes that makes up the endoplasmic reticulum. So let's keep going. Let's have a look of the next one. And what we are seeing here, we're seeing highlighted part of a cell. So the rest has been shown kind of in a dark color. This is going to be our Golgi apparatus. And Golgi apparatus, the description that matches with that is that they regulate the transport and modify new synthesized proteins. So again, that analog Golgi apparatus is a little like a distribution center of a trucking company. So that's what we're seeing there. Looking at the next question, we have mitochondria highlighted. I don't think that that green color is the best highlight, but we'll do with that. And we remember that mitochondria was important for production of ATP. And the process how we made ATP was through cellular respiration. And I'm sure that you've been introduced to that on your prereq classes. If not, I also talk about that on that recording that I made earlier this week. Here we're seeing that endoplasmic reticulum highlighted. So endoplasmic reticulum is important for synthesizing protein, for secretion, insertion into plasma membrane, and lysosomal enzymes. So it really is involved in a lot of processes. So that the proteins and synthesizing proteins at the rough ER is a good clue for this question. Only a few more questions left. Here we have an fill in a gap question, and we are being asked that the organelles that read coded genetic messages, so we're talking about the RNA, and assemble amino acids into proteins, they are called ribosomes. So ribosomes were the structures that converted that RNA into amino acid chain that then became became a protein. And I will continue, but I'll just show it again on the screen since we had this nice description here. So ribosome is this dark blob at the bottom of the screen. It converts the RNA, which is this line, single strand copy of a gene, and it brings single amino acids, connects them together, and once you have multiple amino acids together, they form a protein. So jumping back to our quiz again, the next question that we're gonna see is asking mitochondria's extract energy from organic compounds, so converts sugars, converts sources of energy, and convert it into cellular energy currency, which was our ATP. So ATP is really what drives cellular processes. So the main job of mitochondria is to produce ATP. Here again, we have a little video that I show you first, and you're being asked about exocytosis. In exocytosis, cells release substances via using these vesicles, so a little bit similar as we saw with the lysosomes. And here we're seeing an opposite reaction of that, endocytosis, so phagocytosis, when we're engulfing things into the cell. And again, I'm going to show it just really quickly as an illustration. I'll sketch this for us at the very end. Let me make a blank slide for us. So when we're looking at the scale of a cell, so here we have a cell and we have a nucleus in there. In endocytosis, we have something that we want to get away from the cell, but these little components that we want to get rid of can't just travel freely across this cell membrane. They're not able to do that crossing. So what happens? We put these little elements into a sac, a membrane sac, and that sac then travels to the cell membrane, and there it fuses with the cell membrane. And as it fuses, it's able to then kind of move these materials across this membrane as the sac around the materials fuses with the rest of the wall of the cell. So I always think of it as an endo, as a way to kind of move it, and then we see opposite reaction when we engulf something. That was the phagocytosis. So this is something that we don't want to have floating around in the body. And here we have a cell that eats it. This material enters into the cell. So that was our phagocytosis. So that was really a short explanation. I don't know if it was the best explanation I've ever given. It was the explanation that I could give now. So let's jump back to that question and finish off our quiz. So just to make sure that I got it the right way around. So here we're, I mixed it around, so let me make sure that I pointed out. Exocytosis was what I labeled there incorrectly as endocytosis. So exocytosis is when something is exiting the cell, and endocytosis is when it's entering in. So just to make sure that I did a bit of a bad job there explaining, so let's go back here and just make sure that I correct. So this would be exocytosis. So it's exiting the cell. Think of it that way. Exocytosis, something leaving the cell. And endocytosis is when something enters in. So exo for exiting the cell, endo for entering into the cell. I apologize for, I don't know where my mind was mixing those up initially. Let's wrap up our quiz. So we saw an exocytosis releases substances from the cell via vesicles. It takes them away. Those substances exit the cell. And then phagocytosis is a form of endocytosis, so taking in substances. And the final question, that was the final question, I believe, on our quiz. And that brings us to the end of what I wanted to show to you. I'm just gonna wrap up this session by pointing out that we've covered the homework that I had for this chapter. So if you have any questions of those, you can always look back on this recording, and you have the full review of the chapter material, like I said, on that video that I made on the Wednesday. If you have any questions, I am around here to answer to those. But if you don't have any questions, that's all I have for today. And I'm more than happy also to post the recording if you want to stay behind and ask questions without being on the recording. But that's it for tonight. I really appreciate you sticking around, and sorry about going a little bit over the scheduled time.